Hello, my name is Ling. I will walk you through Module 8 of our synthetic biology class. In this module, we will look at how to write a lab report for biology experiment. The structure I follow here is also used to write scientific papers in journals. Before we start, please open the sample lab report that I've written and open the Google form for your lab report submission from the syllabus. In the first part of the module, we will look at the compositions of a lab report. And in the second part, you will be shown how to do a DNA extraction experiment. The first thing you can write in your lab report is the introduction. Technically, you can start writing the introduction even before you do your experiment. The introduction to tell the reader the purpose of the experiment. What questions are you trying to answer? What hypothesis are you trying to prove or disprove? You should also tell me the context of your purpose. Why do you want to answer that question? In formal lab reports, you will see grand goals like curing cancer or ending hunger, because those authors are trying to attract sponsors and research comrades who will read the lab report because they have the same goals and want to learn from the authors. The experiments you do for your high school and college classes likely aim to teach you some research technique. After the purpose, you should tell me how you come up with your hypothesis. Surely you must have a reason for your assumption, right? The introduction is also where you include all the background information I need to understand why you did your experiment. The depth of detail of the background information depends on your target information audience. Who do you want to read your reports? The general public with minimal biology background would need a more extensive introduction. Your friends with basic biology background but knows little about your particular area of expertise, which I hope is synthetic biology, would need some in-depth knowledge. Your synthetic biology professor would not ask for textbook knowledge, but do not assume they know everything. If you base your experiment on another recently published paper, your professor might not have read that paper yet, so you have to introduce the paper to him or him. Don't forget to cite where you get your background info from. Let's move on to an example where I am a field biologist studying penguins instead of a synthetic biologist. Let's draw a change of pace. My hypothesis is that penguins can smell krills, which are the main food source. This is part of my larger hypothesis that penguins find foods by smelling, which is part of my even larger research into penguin feeding habits. And I want to study penguin feeding habits because I want to use that knowledge to predict if penguins can find food and survive as ocean temperature rises. But why smell? It's been proven that birds can smell things. And I know that krills are smelly, even underwater. And I've read a few papers proving that petrels can smell krills. So I assume that penguins must be able to smell krills too. By the way, you should know that krill smell comes from the dimethyl sulfide that krills emit, because I'm using the dimethyl sulfide, DMS, as the krill smell in my experiment in place of actual krills. Following the introduction is the methods. This section should include a list of materials and equipment and the experimental setup. If you think the materials and equipment can affect your data, make sure to include where you got them, the manufacturer, the product model, and product number. The flask and tubes generally don't get mentioned because there are industrial standards for them, but your cells, the plasmids, the proteins, chemicals, microscopes, and lasers, etc., should all be there. If you use a software to analyze your data, make sure to include the software version and any add-ons you have. For software and tunable equipment like microscopes and lasers, make sure to state the settings you set them to. What magnification was the microscope lens, what wavelength was the laser on, how long was the exposure time, etc. After the materials is the procedure, which are the step-by-step manual on what you did during your experiment. A good rule of thumb is if someone in your lab cannot follow the procedure and redo the experiments, you might need to edit it. Back to our penguin example. I will test my hypothesis by holding a smelly cotton ball in front of the penguins when they sleep and see if that wakes them up. For the materials, I will need some seawater, some DMS, cotton balls, a 2-meter stick, and some glue, and Excel 2016 to analyze my data. And I should tell you that my experiment takes place in a king penguin colony in Antarctica, because maybe other penguins in Antarctica won't react the same way as king penguins, or king penguins in other places won't react the same way as king penguins in Antarctica. For the procedure, soak the cotton ball in seawater or DMS for 30 seconds, glue the cotton ball onto the 2-meter stick, find a sleeping penguin, hold the cotton ball right on top of the beak, not touching the penguin. If the penguin wakes up within 3 minutes, mark as reacted to smell. Otherwise, mark as did not react to smell. Repeat your step 1 with DMS if you used seawater last time, with a new cotton ball, or with seawater if you used DMS last time. Analyze your data with d-test analysis function in Excel. Now we've got to the fun part, where you can share what you find. I've seen people do this in many ways, but this is how I recommend laying out your results. Start by presenting what you find at face value. Remember to present your numbers in words and in graphs. Are there any trends here? Are the trends from your treatment group significantly different from your control group? Now let's interpret the findings. Do the trends agree with your hypothesis from the introduction? If yes, congratulations! I'd love to hear how the result connects to what you previously know about the topic of interest. If not, why do you think your results disagree with your hypothesis? Could your hypothesis have been wrong? Could there be a massive error in your experiment, like mixing up control and experimental chemicals? Could there be an alternative hypothesis that you didn't think of originally? Finally, what are you going to do with these findings? Are you redesigning the experiments to improve on sources of errors? Are you repeating the experiments, maybe with different sample populations or different cell types for more data? Are you using your findings to move on to your next hypothesis? Back to our example. Let's be very positive, and I say that I got phenomenal results from my field study. Only 10% of the penguins woke up from seawater, and 90% woke up from DMS. And the difference between my treatment and my control was significant, because I got a p-value of less than 0.05 when I ran the t-test. This means that there's only a 5% chance that this difference is due to random chance alone, no influence from my experiment from the penguins, like maybe I happen to only catch light sleeping penguins with my seawater and heavy nappers with my DMS, because the chance is only less than 5%, I will assume that this result is not due to random chance, but actually due to my experiment. So I say that the results support my hypothesis that penguins react to DMS. However, looking back, I should have used a dry cotton ball or cotton ball soaked in drinking water as another control, because who knows if the penguins didn't actually get sleepy when smelling seawater instead of getting awoken by DMS, as I'd like to believe. Moreover, if I used the very concentrated DMS I bought from company X straight out the bottle. So I can't say that the penguins can detect DMS from krills when the DMS is diluted in seawater. For my next experiment, I would add another control and another treatment that is DMS diluted in seawater. Wrapping up your lab report is the conclusion. Briefly restate your questions and the answers you get from your experiment. Should the readers be cautious of anything when interpreting your results? Can they assume that they would get the same results with another species or in the natural environment instead of the lab? Things like that. Finally, did you take a step closer to your final research goal with your findings? In my example, I restate my hypothesis and results. I caution my readers against applying my findings to other seabirds because who knows if other seabirds feed the same way as penguins. My finding here will likely make me pay particular attention to smells when I study penguin feeding habits later in life. As I have said in my introduction, I want to predict whether penguins can find food with their current feeding strategies as climate change continues. So my future experiment may be to study whether their sense of smell is affected by rising temperatures. Now that you've had your lab report, it's the time to write the abstract. The abstract always goes on the first page to attract readers, but you should write it last so that you don't miss any important information. The abstract is basically a summary of your purpose, procedure, and results. I will start my abstract with the grand goal of saving penguins from global warming as clickbait. And I parkour from that grand goal to my strangely specific research question and what I did to answer it. Then I gave a suggestion for future research in hope that someone will join me in my research or give me the money to conduct that research. And you've learned how to write a basic lab report. Here's the penguin smell test by Professor Gregory Cunningham for your entertainment. He's from St. John Fisher College, but he teaches an animal behavior class at the University of Rochester. That one trip a bit is funny. Now, you will conduct your own scientific experiment and write a lab report on it. My question for you is, what does DNA from saliva look like? You will want to create a hypothesis regarding physical properties of DNA. What color do you think DNA from your saliva is? What texture will it have? Will we be able to see a lot or a little bit of DNA? Will it be stringy or clumpy? Create a hypothesis that includes predictions to these questions. Then watch the following video, which will walk you through how to extract DNA from your saliva. After that, you will write a lab report about this experiment. Remember to take a picture of the result for your lab report. And that is the end of our lesson. Thank you, and until next time.